Good morning, everyone. Today, I'm going to be talking about soil microbial communities, specifically about establishing trends in soil treated with composted chicken litter. Despite how little the average person might think of it, soil is one of the most important parts of the natural world. Some of the reasons why are obvious, such as enabling us to grow food, but soil can also be a diverse reservoir of microbes that are important to study for their interactions with the environment around them. The thing is, that soil is a pretty complicated medium. It's made up of materials such as humic substances, the result of decomposing plant matter such as lignin, and then also incorporates a wide collection of organic and inorganic matter. Even the things that live within the soil contribute to its complexity. Following this, even just determining the contents of soil and how it changes can be hard. The part that we're interested in specifically is soil microbial communities and how they can be affected by various stimuli. For our purposes, this concerns the introduction of composted manure, which can have a variety of very significant effects on both the soil itself and the communities within it. Other scientists have asked some questions over the years. What do these effects look like, and how long do they last? However, these turn into more complicated questions too. What we're asking specifically is, despite the overall complexity of the soil, are these effects consistent and repeatable? Can they be fully predicted? To take a step back, compost can be made out of many things, but for our study, we use chicken litter in particular. According to a survey conducted by the Department of Agriculture, the United States has over 9 billion farm animals, over 95% of which are broiler chickens alone. On average, for every 1,000 pounds of animal, you'll get about 40 to 80 pounds of manure produced on a daily basis. That adds up to a lot of waste over time, and an effective management strategy is to compost it so it can be spread on pastures as fertilizer. Adding compost to soil is a good way to increase nutrient content, injecting huge amounts of carbon, nitrogen, phosphorus, and other elemental nutrients. However, chicken litter has a microbiome with many kinds of bacteria that you wouldn't ordinarily find in the soil, which means you're potentially adding non native organisms into the environment. Further, the use of prophylactic antibiotics in the chicken industry has led to the development of strains of bacteria that are resistant to certain antibiotics. These things all have a profound effect on soil microbial communities. They are sensitive to the addition of nutrients and other factors, and the introduction of resistant strains of bacteria can lead to the spread of resistance through horizontal gene transfer. This leads us to the foundation of the study we conducted. The composition of the chicken litter microbiome has an array of organisms, but members of the families Lactobacilliaceae and Staphylococcaceae combined made up almost half of the microbiome. Logically, if we were to amend soil with composted chicken litter, we would expect to see an increase in representation of these families in the soil. Based on that idea, we compared soil microbial communities of pastures where some were amended with chicken litter and some were not. Then we analyzed the resultant changes in the community structure with the expectation that amended soil would more closely resemble the chicken litter microbiome than unamended soil. Returning to the questions I posed in the first slide, we come to our hypothesis. Uh, if soil is treated with composted chicken litter, then the resultant effects will be consistent across treated samples. We expect to see an increase in the presence of bacterial groups associated with chicken litter, especially Lactobacilliaceae and Staphylococcaceae. Our objective in the study was to establish more concrete relationships between changes in the soil and their resultant effects. We used metagenomics to determine the content of soil microbial communities, and then employed a number of different statistical approaches to understand and validate the information we obtained. The looser goal with all of this is to pave the way for future studies to determine such relationships in different contexts. Given how complex the soil is, it's pretty likely that studying it will be associated with unforeseen problems or unexpected results. Hopefully, the data and process of the study will help to inform future researchers and allow them to reproduce such studies more easily. Other researchers, both historical and modern, have addressed this topic, but a lot of them only answer it on the side, or with a small number of samples. In contrast, our study sought to answer these questions using a larger scale study, including 16 farms across northern Georgia, mostly in the eastern part of the Piedmont region here. About half of those farms were amended with composted chicken waste at a rate of about 2.5 to 3 tons per acre, and the other half were left unamended. Samples were taken from each farm 2 weeks after the application of waste, and with 5 replicates from each farm, we have a total of 80 samples. As seen on the right, that soil was then sieved with a 4.75 millimeter mesh to remove large debris or other particles, leaving behind fine soil in order to make sure processing was easier and to help ensure that we were only analyzing soil microbes. Sifted samples then underwent DNA extraction. There are a number of substances in the soil that make it difficult to extract DNA from, humic acid in particular being a huge culprit. So in order to make our lives easier, we relied on a proprietary kit. Once we had our DNA, we processed it according to Illumina's MiSeq protocol, which includes 16S rRNA PCR amplification and subsequent cleanup of the 16S amplicon. Once cleaned, the extracted DNA was multiplexed using Illumina's indexing primers and sent off for sequencing at Virginia Tech's Biocomplexity Institute. The DNA sequences we got back were processed extensively using an analytical pipeline called QIIME-2, which stands for Quantitative Insights into Microbial Ecology. As the name implies, QIIME is specifically designed for analyzing DNA for information related to microbial communities. Without going into too much detail, the main steps start with checking the quality of your DNA sequences, which allows you to set standards for what you're willing to allow in the final analysis. In the figure on the right, based on our threshold of 1,000 sequences minimum, we removed 3 samples from the unamended soil and 1 from the amended group. The samples that passed muster were then denoised and filtered to remove sequences from irrelevant taxa, such as from the plant and animal kingdoms. The bacterial DNA left over was subjected to multivariate statistical testing. The most important information we were able to obtain from this pipeline pertained to ecological diversity metrics known as alpha and beta diversity. Alpha and beta diversity relate to different aspects of the idea of species diversity, or richness. As a brief explanation, alpha diversity describes diversity within individual samples, or how many species or OTUs are present. Beta diversity is a measure between samples, describing the similarity or difference in diversity across 2 or more samples. On the right is an example of these properties. Each letter represents a different species or OTU, and you can see in the top box that though it's rather populous, there aren't many different species present, which means it has a low alpha diversity. In contrast, the lower community has several different OTUs present, so it's said to have high alpha diversity. Because the underlying community structure between each sample is very different, these samples together are said to have high beta diversity. In reality, this is a simplification because there are multiple different ways of assessing alpha diversity, and there are typically many thousands of OTUs in soil, which is what makes having a data pipeline so useful. For each of the measures of alpha diversity we subjected our data to, we got a box plot like the one above. In this particular graph, we're examining the ecological idea of evenness, which operates on the principle that communities that have populations with similar sizes have higher evenness and thus greater species richness. As an example, assume a community has 100 organisms spread across 2 species, where one has 90 organisms and the other only has 10. The evenness of that community is low, and thus it is said to have low alpha diversity. Conversely, if a community has 4 species that each are around 25 organisms, then its evenness is high, and correspondingly it has high alpha diversity. The evenness score is represented as a value bound between 0 and 1, where 1 corresponds to high evenness. As it is, between the untreated and treated testing groups, we saw an evenness score near the upper bound in each community, and as such there was an insignificant difference in alpha diversity by this particular metric. As it is, evenness was actually the measure of alpha diversity that was closest to being significant. The others are far less so, which is highly indicative that there was a similar level of species richness between both testing groups. To go into a little detail, other metrics we considered included Faith's measure of phylogenetic diversity, which accounts for the number of clades present in a sample as well as their phylogenetic distance from each other. OTU count is what it sounds like on the box, where a higher number of species in a sample confers higher alpha diversity. Shannon's index is a combination of the concept of evenness and then the abundance of the species within samples. A possible explanation for the similar alpha diversities in both testing groups may be found in the underlying similarities between them, by which I mean the fact that all the soil samples from both groups were taken from pasture. It's been found in other studies that cropland and pasture are some of the most diverse environments on a microbiological scale. Even with the addition of composted chicken litter to some farms, seeing similar levels of alpha diversity between them makes some sense. Now then, we turn to beta diversity. As a reminder, this ecological metric is about comparing underlying community structures between treatments. Using multivariate nonparametric analyses, it's possible to take an enormous amount of complex data, often consisting of hundreds to thousands of dimensions, and collapse it all into only a handful. These dimensions can then be turned into a scatter plot, which reveals information about how each sample is related to each other. We used three methods for analyzing the beta diversity of our samples. Unweighted unifrac takes into account only the presence of sequence variants and their phylogenetic relationship to each other. Weighted unifrac does this as well, but also factors in abundance. Finally, Bray-Curtis looks at the presence and abundance of sequence variants, but it does not account for the phylogenetic relationships between them. Using all of these in conjunction with one another can help to paint a better overall picture of what's happening between treatments. In these scatter plots, each one of these dots corresponds to a sample taken from the soil, so there's 42 points here for unamended communities and 34 for the amended. As a reminder, these visualizations are a simplification, or a reduction in the complexity of the original dataset. If each sequence variant found in a sample is considered a dimension, then you're simplifying all of those dimensions into just the two seen here. The percentage by each axis describes the amount of variation that dimension is responsible for. Obviously, these two axes don't add up to 100% of the total variation, but they were the most impactful. The unweighted unifrac scatter plot shows a huge degree of overlap in communities between each treatment. As stated during the explanation of alpha diversity, despite the addition of chicken litter, the underlying medium is still pasture soil in both treatment groups. Thus, the large degree of overlap is somewhat reasonable. It should be noted that, despite the overlap, the communities in amended soil appear to be more clustered together as opposed to unamended samples. This could be indicative that amended communities have more similar composition to each other than unamended communities. I mentioned before that we reduced the data down to a handful of dimensions, not just two. This is the same form of analysis with dimensions 1 and 3. You can see that dimension 3 has less influence than dimension 2 by a little under 1%, but the corresponding visualization is fairly different, showing a little more of a shift than dimensions 1 and 2. For each ordination, we were able to simplify the data to 5 dimensions using whichever combination of those dimensions we so chose. For simplicity's sake, though, I've chosen to keep the main presentation to only the first and second dimensions for the other two visualizations, with the first and third dimensions for the other two analyses available in supplementary slides. Moving on, weighted unifrac takes phylogenetic relationships, the presence of OTUs, and their abundance into account. The clustering effect seen previously is more pronounced here, and the shift between treatment groups has become somewhat more distinct. Given that this method also takes into account abundance, this is an indication that the chicken litter is causing certain types of bacteria to become more common in the amended soil communities. The Bray-Curtis plot shows by far the greatest separation between the amended and unamended communities. There's no clustering effect here, which is probably due to the ordination not taking phylogenetic relationships into account. Seeing as Lactobacilliaceae and Staphylococcaceae account for almost 50% of the chicken litter microbiome, soil that's being exposed to composted chicken litter should ostensibly see a shift where those families are more represented in the community. So, taking all of these graphs into account, it is certainly clear that there is a shift in the soil microbial community once composted chicken litter is applied. So far, we've been talking in qualitative terms, but of course we want to make sure that this is statistically sound too. We used analysis of similarities, or ANOSIM, and permutational multivariate analysis of variance, or PermaNova, which is a mouthful, to take the sequence data and concretely assess the dissimilarity between testing groups. ANOSIM is represented as a value bound from 0 to 1, where a value closer to 1 means higher dissimilarity. PermaNova is represented as a ratio, where higher ratios mean higher dissimilarity as well. The test described that there was some dissimilarity between treatments, but not an enormous change. Most importantly, across all these tests, there was a highly significant difference between untreated and treated communities. From this, we've determined that there is indeed a shift, but now the question is, what exactly is being shifted and just how pronounced is that shift? In order to figure out, we analyzed the sequence data and assigned taxonomy. To determine how a given taxon shifted between treatments, we subjected the samples to analysis of composition of microbiomes, or ANCOM, which generates volcano plots that detail how much a community changes between treatments. It uses pairwise comparisons to see if two or more communities appear to be different from each other, and if they are deemed different enough by the algorithm, then the null hypothesis is rejected. ANCOM doesn't utilize p-values, but if the method determines that enough null hypotheses are rejected for a given taxon, then it's marked as notable. Using ANCOM, we're able to plot the presence and frequency of certain taxa, and we did this from the phylum down to the family levels. Although the phylum level is too superficial to make any real conclusions, it yielded pretty simple results, so I'll use it here as an example of how the ANCOM volcano plot is generated and what it means. SIR on the x-axis refers to mean abundance, and its absolute value is an expression of the extent of a shift. The W statistic on the y-axis is an analog to significance, with a higher W statistic suggesting greater significance. Each dot on the plot represents a single taxon, and to be clear, these darker dots on the bottom are just the result of multiple dots sitting on top of each other. This taxonomic level had a pretty simple but pronounced shift. Firmicutes was far and away the only phylum that saw a change between unamended and amended communities, which makes sense given that the families we expect to see in amended soil fall under this group. In order to gain a better understanding of what's actually shifting around, though, we need to go down to a lower taxonomic level. Based on the prevalence of lactobacilli and Staphylococcaceae in chicken litter, we might expect to see a significant shift in their respective orders, Bacillales and Lactobacillales. However, the only order that ANCOM determined noteworthy was Tauriscibacterales, of which the genus Tauriscibacter is loosely associated with gut illness in chickens, which might explain its presence in the soil. Bacillales is demarked here and does see something of a shift, but Lactobacillales didn't see much of a shift at all. There's a lot more evidence of shift on the family level, but some of it is a little unexpected. The only family that was marked noteworthy by ANCOM appeared to be this unclassified taxon in the phylum Chloroflexi. The fact that there's no type strain available for this taxon doesn't really leave much for us to go on, so I'll avoid making guesses as to why it's located in our samples. More relevant to our study, Lactobacilliaceae and Staphylococcaceae weren't actually detected in our samples, amended or not. In order to get a better grasp of what was going on, we were also able to develop taxonomic summaries for a more quantitative understanding of these changes. In these, based on sequencing data, it is determined how much of the community a given taxon takes up, represented by cumulative relative abundance, which is equivalent to the percentage of a community that a given taxon makes up. The top 9 most abundant taxa for each level were considered, and all others were collapsed into an others category. On the order level, we see that Bacillales is represented here, but contrary to ANCOM, sees a massive increase in amended soil, but Lactobacillales wasn't abundant enough to make the cutoff into this chart. On the family level, we see a more similar development to ANCOM. Though there are other families significantly affected, Lactobacilliaceae and Staphylococcaceae were not represented in the relative abundance of these soil communities. Bacilliaceae is by far the most heavily shifted, with this massive increase in relative abundance in amended communities. After this somewhat unexpected result, I decided to look at the raw data, which was even more striking. Bacilliaceae experienced nearly a 2-fold increase in relative abundance, but Staphylococcaceae and Lactobacilliaceae weren't even detected in their soil samples, not even in the amended group. Given the relative abundance in chicken litter, the complete lack of any indication of the presence made it clear that there was more going on here than simple deposition of bacteria into the soil. Our hypothesis at this point has already been supported, in the sense that we've seen a distinct shift in soil microbial communities once they're amended with composted chicken litter. However, now we have a couple of new questions. The first of which is, why are we seeing this massive increase in the family Basilaceae? There are a couple of possible explanations, but first it's prudent to return to basics. Soil microbial communities are affected by a wide range of factors, such as temperature, oxygen, and water content, but studies show that the most important one by far is acidity. In our situation, chicken waste is very high in urea content. Urea decomposes into ammonia through microbial processes such as the action of the enzyme urease. Ammonia reacts with water to form ammonium and hydroxide ions. The increased concentration of hydroxide caused by chicken litter can increase the pH of the soil as high as 9.0. This matters because Bacillus species have an ideal growth range of 7 to 8, which would mean that the addition of chicken litter is potentially making the soil more habitable for members of Bacilliaceae. Based on this, we decided to run an electrometric assay to determine the pH of each sample. The average pH of unamended samples was 5.67, and for amended communities it was 5.91. At first, that seems like a pretty low increase, but due to the logarithmic nature of the pH scale, it actually comes out to a 173% increase in the concentration of hydroxide ions for amended soil samples, which corresponds nicely to the twofold increase in Basilaceae. This may not be totally convincing, given the fact that the average pH is still below the ideal range of growth for Bacillus species. As I described at the beginning, though, soil is a really complicated medium, and part of that complexity manifests in the idea of heterogeneity. It's possible, or even likely, that soil amended with litter saw the formation of little pockets where the pH was much higher and facilitated the growth of Bacillus species. As it is, though, this by itself isn't enough to explain the lack of growth in Lactobacilliaceae and Staphylococcaceae. Members from all three of these families share at least some commonalities in preferred pH, temperature, and the ability to respirate, so there's probably something else involved here. Thus, our second question is, why exactly do we not see an increase for these groups found in chicken litter? As a reminder, Lactobacilliaceae and Staphylococcaceae make up almost 50% of the chicken litter microbiome. In contrast, the relative abundance of lactobacilli was less than 0.5% in amended soil communities, and Staphylococcaceae weren't detected at all. In order to puzzle this out, we looked at different properties of the compost litter and its application, such as how much was applied as compared to how much soil was sampled. There was absolutely a higher ratio of soil to litter, but only by a couple of orders of magnitude, whereas both types of media typically form 10 to the ninth colony-forming units per gram of material. Additionally, the method of application affects how much interaction there is between litter and soil. In our experiment, litter was spread onto pasture by broadcasting, which is simply throwing it so that it rests on top of the ground. It's possible that we would have seen more bacteria from the litter if it had been applied by other methods like subsurface injection. Another angle to answer this question lies in properties of the composting process itself. The chicken litter microbiome doesn't necessarily correspond to what's being put in the soil, because depending on how material is composted, it can result in a couple of effects. A pretty common effect is that composting will reduce pathogenic load through thermal action, and certain kinds of bacteria in litter may be reduced to the point that they are undetectable. Conversely, in some techniques such as static windrowing, the population of Bacillus species is massively increased. Through this, we've obtained a possible explanation for both of our questions. The composted chicken litter probably added to the Bacillus populations already in the soil. Conversely, due to factors such as the ratio of litter to soil, the method of application, and the reduction of pathogenic load, it's likely that the bacterial groups from the chicken litter never made it into the soil to begin with. As such, their respective increase in undetectability are actually fairly reasonable. So, to recap everything, measures of alpha diversity indicated that there was an insignificant difference between samples, which is reasonable given that the underlying media was similar between testing groups. Multivariate scatter plots and statistics show that beta diversity was significantly different, suggesting that chicken litter was causing shifts in the composition of the communities. The precise shift we observed was a twofold increase in Basilaceae rather than in groups common in chicken litter, but based on soil conditions and especially the process of composting, this is a more or less explainable result. From all of this, the stated hypothesis was supported. We witnessed a consistent and repeated shift in soil microbial communities based on the amendment of composted chicken litter. In order to further validate this study, obviously it needs to be repeated. In particular, there are two more time points in consideration here. The second time point was sampled 4 months post-amendment and the third at 6 months. In addition, it would be prudent to analyze other facets of soil microbial communities, especially fungus, using ITS sequencing. This is a priority for me personally due to arbuscular mycorrhizal fungi, which is enormously important for the uptake of nutrients from the soil. Further downstream, it would be good to conduct more large-scale studies like this related to strictly abiotic factors such as pH, temperature, and water content, and then later perhaps other specific treatments of the soil. For example, this study was only based around the application of composted chicken litter to pasture soil. Other possibilities include other kinds of animal waste, other soil types, local vegetation, and so on. To bring this back around to the start of the presentation, eventually data like this could be used to develop reliable models for predicting changes in soil microbial communities caused by introduction of antibiotic resistance genes. This could be very useful in the fight to prevent proliferation and spread of resistant strains of bacteria that threaten the medical world. For all of this, I'd like to acknowledge my colleagues in the Williams Lab, especially Hazem Sharaf, who developed the data pipeline we use in this analysis, and Grace Carey for conducting a great amount of the wet work. I'd also like to thank my committee members, Translational Plant Sciences for giving me the opportunity to conduct research at Virginia Tech, and the USDA's National Research Initiative for funding this research project. With that, I'll take any questions.
Okay, good. Thank you, Elin. Yeah, please, anybody have any questions? Go ahead and speak up.
So I have a question, Elon. I remember going through your thesis, and then you mentioned it, but in passing. So is there anything to the differences in the Clostridiales in the amended versus unamended soils?
Yes, actually. I have that up here. Clostridiales did experience a slight increase and is in fact one of the few that we saw a lot in chicken litter that experienced a significant increase. Uh, so there could be something going on there.
Is that an order that's been reported in soils, especially pasture land?
To a limited degree, yes. Not overly much, and it's an enteric organism, so it makes sense that it's not highly represented there. But soil is pretty complicated, so it makes sense that we'd see at least a little bit in the soil.
Okay, I'm sure we have another question out there.
Elin?
Go ahead. Sorry.
Oh, I'm sorry. Hello. Elin, so where do you think the Lactobacilliaceae and the Staphylococcus went? I mean, it was undetected. So how does that break down exactly? What are your thoughts on that?
Well, like I said, it's possible that they just never made it into the soil to begin with. But if they did, then it's important to keep in mind that this soil was sampled 2 weeks after the application of chicken litter. So it's possible that they degraded over time, not degraded, excuse me, but their population saw a reduction over time. Studies suggest that compost amendments to the soil, in terms of microbiological impact with the certain species that you see, don't persist over time. They actually tend to reduce pretty quickly. So I wouldn't be surprised if that's what's happening here, that they just naturally reduced over time.
Okay, thank you.
Okay, any other questions out there? I'm sure somebody has something else that they're curious about.
I have a question. I really don't know much about the soil organisms, but I'm kind of wondering about the lactobacillus. So, do any members in the soil have any nutritional requirements, amino acids or vitamins, or most of the microbes in the soil thought to be capable of making some of these components?
Well, the soil microbiome, uh, well, it's a simplification to say that it's outrageously complicated. Um, Not only does it vary within a particular environment, but you've got so many different types of soil out there. So, in the broad sense, to answer your question, yeah, I'm sure that there are organisms in the soil that require those vitamins and nutrients that you're talking about. But they're probably usually there at like a really low level, a baseline level, based on what's available. So, um, with the addition of chicken litter, compost chicken litter, even that probably changes quite a bit.
Yeah, the reason I asked is because outside of Lactobacillus plantarum, most of the Lactobacillus species that you would find in the intestine, regardless of whatever animal species, tend to be auxotrophs and have some sort of amino acid or vitamin requirement. We think they're auxotrophs because they don't need those anymore because they can get all their nutrients from their animal host.
Makes sense. Yeah, I mean they are usually enteric organisms, so that makes complete sense.
So we have another question here online that I'll ask. Are there other types of manures from different animals known to change the pH more drastically than, say, chicken manure or chicken litter?
Well, that's an interesting question, actually. Uh, Chicken litter in particular is high in urea content. Obviously, nitrogen forms of nitrogen are also present in cattle and pig manure. I don't know if it's substantially less than chicken litter, but I imagine that there are some compositional differences based on gut microbes and their diet that would change how they affect the soil microbial community.
Sort of as a follow-up, because you mentioned models, the question, kind of follows up and asks, considering the potential for other amendments to be potentially similar, could you use other amendments, or have you looked at other amendments in the literature to perhaps might support this type of work, whether it be cattle manure or turkey litter? Um, have you seen any of that, and has any of that been discussed, or do you
Uh,
think this? Yeah, go ahead.
no, yes, I've looked into it a little bit, not extensively. As far as turkey litter is concerned, poultry litter, it does experience some differences, but turkeys and chickens are certainly closer to each other than chicken and pig or chickens and cows. Uh, as far as other studies extensively looking into this, I'm not extensively aware of any. I'm sure that they exist, but I haven't had a chance to read them personally.
Yeah, that's a good question there from Catherine, and I think that's something you could look into to sort of support your model, or certainly undercut it one way or the other. So I think that's a good question.
Thank you.
And I have actually one more question. Oh, and great talk, Elon. I really enjoyed this.
Thank you.
I was going to say, so do you know exactly what type of like chicken that this manure came from? And if so, did you would it be possible to like compare the microbiome of the specific like type of chicken to like what you're seeing in the soil?
Yes,
Or
actually. All of this chicken litter came specifically from broiler chickens, which are grown
oh, that's
grown specifically for their meat. They are turned into meat products. If you were looking at hens instead, which are used for egg laying, you would probably see a difference because the composition of their manure is slightly different. Higher water content, for instance, the water content of broiler chickens is at 25%, and for hens, it's at 35%, 37% or so. So from that alone, not even to speak of the nutrients, you would probably see a pretty big, well, not maybe big, but at least some difference probably in the effect.
Okay, good. All right. Last chance for maybe one more question out there. If not, we're going to end the seminar. All right, everyone. Looks like we're already getting some good feedback from the audience. From there, we can.
Oh, Sophie has one question.
Oh, there we go. Okay. Go ahead, Sophie.
Do you expect Basilaceae to remain high at the other sampling time points you post in your future directions? No, because...
Can you repeat that, Elin?
Oh, sorry, I do mumble, but do you expect Basilaceae to remain high at the other sampling time points you post in your future directions? And the answer to that question is no, because I mentioned earlier that the effect of compost tends to not be very long-lived. As far as the bacterial component is concerned, I read a paper recently that suggests the physiochemical aspects of litter are what tend to affect the soil microbial community the most. And if the deposition of bacteria is mostly associated with Basilaceae, then both between the fact that the microbiological component doesn't have that much an effect and the diminishing nutrients over time, I would expect them to decrease significantly at 4 months and especially 6 months.
I see. Thanks.
No problem.
All right, Dan. With that, I think we will go ahead and end our this part of Elin's defense. Please give a round of applause virtually.